Photosynthesis is the base on the life on the Earth, and this process is based on the conversion of low-energy inorganic molecules into high-energy organic molecules, and it's driven by the solar energy. Two phases can be distinguished, the light reaction and the dark reaction, but those will be explained in details later. Natural energy sources are very different. What photosynthetic organisms use for their biochemistry is the solar energy, derived by the sun, and this is an almost unlimited source of energy. What we call light is an electromagnetic radiation with a specific wavelength range. All the electromagnetic waves are characterized by a wavelength and a frequency, and this parameter provides us information about their energy level. The light is the electromagnetic radiation between 400 and 700 nanometer, and in this range is visible to the human eye. The part of the spectrum used by the photosynthetic organisms is the range between 400 and 800 nanometer and is defined PAR, or photosynthetically active radiation. As we said, photochemistry converts inorganic molecules in organic molecules using the energy of the light source. In photosynthetic organisms, the light harvesting molecules are the pigments, and the absorption of the light energy produces an excited state of the pigments. Once excited, the energy absorbed is used to generate a charge separation between the pigments and the acceptor molecules, with the transfer of one electron to the acceptor. After the charge separation, the pigment takes an electron from the environment, restores the depletion of one electron. Pigments are divided in two groups, chlorophylls and carotenoids. They have different absorption area in order to cover almost the entire spectrum of the light. Furthermore, carotenoids have also the function to protect organisms from too high light intensities. Photosynthesis is present at different evolutionary levels. In plants, of course, but also in the aquatic environment with macro and microalgae, like the two species here represented, widely used in research. Photosynthesis occurs at prokaryotic level too, for example in cyanobacteria or purple bacteria. In this picture is represented a plant at four different levels: macroscopic, cellular tissue, organelles, and thylakoids. The compartment of the cell where photosynthesis occurs is the chloroplast. Chloroplast is an organelle surrounded by a double membrane that encloses the stroma. In the stroma there are different enzymes and ribosomes for the synthesis of photosynthetic proteins. There is also the chloroplast DNA, derived by the endosymbiotic event. Into the chloroplast we find a 3D network of membranes, in which are embedded the photosynthetic complexes for the light-driven reactions. This membrane network is called thylakoids. The thylakoids are divided in two types of membrane, grana and stroma lamellae. The granum is composed by several discs stacked together, and these stacks are connected each other by stromal membranes. The area surrounded by thylakoid membranes is called lumen. The organization described before is just one example of how the chloroplast and the thylakoids are organized. Different structure can be found in nature, comparing photosynthetic organisms at different evolutionary levels. For example, in cyanobacteria, thylakoids surround the entire cell. In Chlamydomonas, a green alga, they present this peculiar structure, or in the diatom Phaeodactylum tricornutum, they have four membrane envelope due to the origin by secondary endosymbiotic event. It is important to underline that function and structure are connected in the chloroplast. So different shapes and structure correspond to different regulation mechanisms of the photosynthetic apparatus. So we can conclude that the function and the structure are strongly interconnected in the photosynthesis.